that progresses. So we'll start off. So this is our agenda for today. So we'll have an introduction and overview to Shannon ABC, which will include a virtual tour of some of the laboratories that we have both in Tralee and Limerick. We'll go through an overview of funding mechanisms, and then we'll have some industry case studies of companies that we worked with, both from the company perspective and then from a technical perspective in terms of how we actually carried out the work. Towards the end of the event then, we'll have our showcase video clip, which we're very keen to present to people. And then at the very end, then we'll have our Q&A session. So again, if people can hold off on questions till then, that'd be super. So just a bit about Shannon ABC in the first instance, I suppose. So we're a commercially focused research center and technology gateway. Here's some stats, I suppose, about what we've done over the last number of years. And certainly one of the things that we're very keen to point to here is kind of the range of projects that we engage in. So from very short projects for a couple of days to very long multi-partner projects. And again, in terms of value, you know, we're very happy to do small projects with companies where it's required. It's not a prerequisite that a project should be in the thousands for us to help companies. Fundamentally, we're here to help Irish companies, and however we can do that, that's what we're here for. So we're a collaboration between Limerick Institute of Technology and Munster Technological University. And hopefully a lot of you may be aware of the fact that Limerick IT have recently been given approval with Athlone IT to become the Technological University of the Shannon, Midlands, Midwest. And so from, I think, the start of October, Limerick IT and Athlone IT will be officially TUS. So we're looking forward to that. So Shannon ABC is a technology gateway, and this is the technology gateway network. So there's 16 technology gateways based all around Ireland, and each of the gateways has their own area of expertise, but they all operate in more or less a similar fashion in that they have business development staff, project management staff there to make sure that all the projects with industry run to plan. So this is us here, Shannon ABC. So we're the only collaborative technology gateway, the only gateway based across more than one college. And we certainly feel that brings some benefits to companies that we'll talk to later on. But I suppose a big advantage of this network really is that you can talk to us about any particular project. And even if we can't do it, we'll know somebody within this network that we can introduce you to who will be able to help you with your project. And so it's really important that even if you think the particular research area that we don't work in, please do feel free to contact us and say, look, I need to find somebody who can help me with photonics research or precision engineering or wireless technologies or whatever it is. And we'll be able to point you in the right direction of who you should be working with. So what does Shannon ABC do? So this is a big kind of wordy statement, but really I suppose the important word here is bioresources and helping companies and organizations get more value from bioresources. And whether this be as a raw material or as a byproduct, that's really why we're here. And I suppose we've built up quite a lot of tangential expertise that we can help companies with, but primarily we're here to help companies get more value from bioresources. And so how do we do that? Well, we've got some very nice laboratories, both between Tralee and Limerick, and this takes a couple of seconds to load. So we'll be showing you the labs that we have, but it's about probably 50% of the laboratory space that we have and equipment that we have, but I didn't want to be going through it in too much detail, I suppose. So these are the laboratories in Tralee. So this is the main lab that we have here. To the left, as we're walking through, there's another large laboratory, but we'll just show you the main lab that we'll go into here now on the right-hand side in a moment. So we have a good bit of space here. We have companies who use this facility, and there's Soji with his mask working with a postgraduate student. So we have an autoclave room there. We have a cold room at the back right there. I realize we're going to a little bit quickly maybe. So we've got a nice bit of lab space here, which is good because of COVID. We need to socially distance quite a lot. In Shannon ABC in Tralee, we do a lot of work with cell culture. So growing mammalian cells in flasks and seeing what happens when we introduce different extracts or products into them. And so this is one of our cell culture suites. So we've got 3 hoods in here that we can work with cell culture material. We have a flow cytometer to help us count cells. And we've got different incubators, both shaking and static, for growing the cells in. So that's one cell culture suite that we have. There's another one across the way here. And as we go across, we'll see we have a plate reader here on the left-hand side, real-time PCR as well, and another incubator for growing different types of cells. This is our second cell culture suite. This has, I think it's 2 or 3 hoods in here as well for us to work with different cell culture materials. So it gives us a lot of capacity to work with companies. And as we pan around here, we'll see this is our rocking platform for a new single-use bioreactor that we got funded recently, which we're very looking forward to get working on. So we have a postgraduate student who's recently started in a project to look at the comparison between single-use and stainless steel bioreactors. This is our storage room, so -80 freezers in here. And then we also have a freeze dryer here that we got funded through a recent Enterprise Ireland call. And so we've been putting that to good use recently for a number of different industry projects. This is some of the laboratories in Limerick. As we come into the labs in Limerick, all our labs are closely controlled, so it's swipe card access to all the different laboratories. And so as we go into the lab here, this is kind of the preparatory lab here, I suppose, to allow us access to work with different materials. So we've got a fume hood there. This is our microbiology lab, and there's Mariana working away. So we have two class 2 hoods in here. We have a bioreactor facility down the back there, small scale. And then we also have real-time PCR in LIT as well. We have a freeze dryer here that we'll pass on your left-hand side. And then we have a liquid handling robot as well that's available for use. So in LIT, analytical chemistry is a very strong area for us, and being able to identify a range of different types of compounds. So it's a little bit noisier in this lab. So this is our LCMS. And as we start to walk through the lab, we have a spray dryer that we can work with companies on, looking at concentrating down different liquid materials. As we walk through the lab here, we have our high performance thin layer chromatography, which is a new piece of kit that we're getting more use out of at the moment for identifying different compounds. We also have a large scale centrifuge for centrifuging large volumes of liquid, supercritical fluid extraction, and our new triple quad liquid chromatography. So we also have other liquid HPLC. We've got atomic absorption. We have GC. We have GCMS, which you'll see as we pan around to the left here. So in essence, any compound that we want to detect and identify, we can. So I say very nice analytical suite in Shannon ABC in Limerick. Now, obviously, we need somewhere to actually do the work to produce the materials to test on that equipment. And that's what we have here in our wet lab. So we've got space for 8 researchers in here, and that's Biba working away down the back of the lab. And so this is, as I say, is where the researchers can actually work in a little bit more peace and quiet to get their samples ready to work in the analytical suite. So our final video that we have is of our photosynthetic growth chambers. So we have plant growth chambers in Limerick as well. We work with the cells research group in terms of looking at different endpoints of plants and how different extracts may impact those in terms of things like germination rate, shoot height, leaf width, productivity of biomass, and that kind of thing. So these chambers are controlled temperature, humidity, CO2, and so we can mimic lots of different environments in the world to see how it impacts on plant growth. And so this is just an ongoing experiment here, where you see these little pots here all contain individual plants with a different type of growth medium there to determine what the impact is on those in terms of their growth rate. We also work with microalgae, which are bacteria-sized versions of seaweed, and they also use light to grow and different types of light. So you see these different colors, LED lighting here will induce the microalgae to grow in different ways. And if you want to, I suppose, discover any more about microalgae and what we do with them, would encourage you to look at our website for more information about those. So that's our kind of a virtual lab tour. And as I say, I suppose, be conscious of emphasizing that it's about half of the facilities that we have. And once COVID has gone, or at least once it's abated a small bit, we'd certainly be delighted to have you down to the labs to show you around some of the other facilities that we have. So going back to what Shannon ABC do, so fundamentally our work is divided into kind of 4 broad areas. So bioprospecting, which is looking for high value compounds. Bioprocessing is the scaling up of those high value compounds. Analytical and research services is everything else around that in terms of proof of concept and how things actually work. And then food innovation. I suppose food innovation is important because we've always worked with food companies because of what we do. And it's important that's called out to a certain extent so that these companies are aware that we can support them as well from ingredient to final product. So some of these pictures will be familiar from the lab tour that we just looked at there, but just to give a bit of context in terms of how Shannon ABC work with companies in terms of processing samples and how we actually do that. So we need to extract what's in the biomass in the first instance, and that might be through a different number of different types of methods. We need to concentrate that down to be able to work with it. So we've got a spray dryer, freeze dryers, and rotary evaporator. And then we need to identify the compounds, because the real value in terms of a product or what it is how, if you know what the actual active ingredient is. And as I mentioned, we've got a very nice analytical suite to be able to do that. And then finally, efficacy and mode of action. So how does the extract actually work? And for that, we use our cell culture facilities that you saw, we use our microbiology facilities that you saw, and we also have skin probes for cosmetic testing that can be used to look at mode of action and efficacy of different types of products. So there's a wide variety of different routes an extract can take, I suppose, in terms of us determining what's in it, how much is there, what it is, and what can be done with it. So who we are. So what's really important here, I suppose, is we've got a wide range of scientists working in Shannon ABC. So from molecular biologists to microbiologists to fungal specialists, bioeconomy specialists, analytical chemists, a wide range of researchers. And what's important there is that it's that connection between all those different types of researchers with different backgrounds that help us to solve the problems for industry clients. And I suppose the next slide in terms of the types of companies that we work with goes back to that kind of broad base that we have around bioresources. We work with companies across all these different sectors. And what's important about this is that if we're looking at a byproduct from the food sector, for example, we're not necessarily looking to put the results from that back into the food sector. We're also looking at marine and cosmetics and nutraceutical, for example. So that broadens the horizons of the companies that we work with and gives us all more opportunity to commercialize different types of bioresources and to get more value from them. So that's kind of broadly speaking around Shannon ABC. What we're here to do is to work with companies and help the commercialization of Irish research. And the way we do that, I suppose, is that we've got a business development team of myself, Elva, Neil, and Lena. And then we're supported by the wider Shannon ABC research team of postdoctoral scientists, research assistants, and postgraduate students that are essential for the delivery of the projects. And so with that capacity, we would talk to the companies around what it is they want to do. Certainly a lot of what we do is help companies identify different funding options, because based on what we have, we can do pretty much whatever you want, but a lot of it comes down to budget. And so it's really important for us to work with companies to try and identify how different types of research can be funded. And so I'll talk through some of the options that we would discuss with companies now, and then you'll see some examples of those as we move on to the case studies later on. So there's a range of different funding mechanisms. Certainly Enterprise Ireland are very active and very supportive of the research that's done with Irish companies and would fund a lot of the different types of options. The first that we do have is contract research and direct funded. And while I suppose that can often be the lowest priority from a company perspective because it's money coming out of the company and isn't a funding mechanism as such, we do emphasize that we're more than happy to work through contract research and direct funded projects. And certainly for some of the time, a company will actually preferentially want to work through contract research because it makes the management of the intellectual property that slight bit easier, because at the end of contract research, certainly the majority of the time, the IP belongs to the company. And so sometimes company will opt for contract research over other types of mechanisms because of the treatment of the intellectual property. The way that we approach contract research from a practical perspective will be the same as all the other projects. We agree a set of deliverables with the company. We'll sign a contract based on those deliverables. And then the report that we provide is based around those deliverables put into action and then put into paper when we have the data from it. We work with a wide range of sectors. I mentioned some of them earlier on. I suppose the link between all these different sectors is bioresources and the capability that we've developed based on our expertise with bioresources. Innovation vouchers are very popular with companies. So Enterprise Ireland funds the Innovation Voucher Programme. And any SME in Ireland, more or less, is eligible to apply and be funded with an innovation voucher. You have to be a limited company. Sole traders can't apply for them, but once you're a limited company in Ireland, you should be able to apply for an innovation voucher. And the function of these is to try and help companies answer specific technical or innovation challenges. They're not intended for things like market research, for example, because there's private companies who do that kind of work already. Any SME is eligible for 3 innovation vouchers. So 2 vouchers worth €5,000 and 1 co-funded voucher worth up to €10,000 that Enterprise Ireland put in half and the company puts in half. VAT is required to be paid on the vouchers, and the intellectual property at the end of the vouchers is treated as per contract research, in that the company will own the IP at the end of the project in most cases. The innovation voucher call is open all year round, so you can apply for whatever. It's a very straightforward process. If you Google Innovation Voucher Enterprise Ireland, you'll get the link. You create a username and password, and then you can fill in the online form. It should take an hour or so. And we're more than happy to review finished application forms the companies have put in, just to make sure that, just to kind of sanity check them to a certain extent. Innovation vouchers can often lead on to longer term partnerships and larger projects, and it can be oftentimes are there to kind of de-risk those larger projects, or they may simply be there to answer a specific question from start to finish. The feasibility study, so the next stage of funding up from an innovation voucher is called an innovation partnership, and innovation partnerships will fund between kind of 30 and 300,000 euros worth of a research project, I suppose. In order to de-risk that larger innovation partnership project, companies can apply for what's called a feasibility study. And so if there's a specific part of the innovation partnership that you're not sure about as a company and your research provider isn't sure if it will work or not, you can do a feasibility study to de-risk that or to create a body of knowledge to help support delivery of the innovation partnership. Feasibility studies are funded up to €9,000, and that's fully funded by Enterprise Ireland, direct to the research providing organisation, or RPO. And that will be done in collaboration with the partner company. So as the feasibility study finishes and you've de-risked your innovation partnership, or you've created that body of knowledge, you can apply for an innovation partnership. And as I say, these are the much larger style projects, generally last from anywhere from 6 months to 24 months, and answer kind of a reasonably significant challenge. Enterprise Ireland will fund up to 80% of the eligible costs of the project, depending on the size of the company and the type of research. And it's very important to consider that. Companies at very early stages of development are generally not eligible for innovation partnerships, but it's certainly worth a conversation with Enterprise Ireland or your state agency advisor to demonstrate that that's where you want to go to. We do a lot of innovation partnerships, and we're very happy to talk to companies about how they might fit into their innovation pipeline. So I suppose the important thing for the innovation partnerships at the end of that project, because the state has majority funded the project, the intellectual property belongs to the college and they license it back to the company, depending on what the company wants to use from that project. So there are other types of funding support as well, and I think this is the last slide in funding support. So Horizon Europe is one that gets a lot of press in terms of the amount of money that's available there. Shannon ABC, we do work in Horizon Europe as well, and we submit and secure different types of European funding. We're very happy to talk to companies about how they may want to be involved with Horizon Europe and to provide suggestions around that. Depending on the type of call that's submitted, the company can benefit specifically from Horizon Europe, which isn't the case for all these types of funding mechanisms, but from Horizon Europe it is. The Disruptive Technologies Innovation Fund is again one that companies can benefit from directly, and indeed you don't need a research performing organisation to be part of your consortium for the DTIF fund, but obviously as Shannon ABC, we'd certainly hope that you consider us for that. So if you want to find out more about the DTIF fund, if you look at the Government of Ireland website around that, they can give you some more information. We have experience of submitting applications for that, and if you do want to have a discussion around that, we're happy to do that also. Science Foundation Ireland have a range of different types of industry-focused research funding. In general, it's around placing different types of researchers in companies in collaboration with an RPO. So the company wouldn't benefit directly financially, but certainly could benefit substantially from having somebody on site who's very significantly experienced in their research field. Again, the Irish Research Council fund collaborative postgraduate and postdoctoral scientists between industry and academia, and they're an excellent program to look at. And Interreg Europe is another European funded mechanism that can be appropriate for different types of industry projects. And I suppose really why they're here is just to give you an outline of the different types of funding that are available, and that if you're interested in talking about different types of projects and different sizes of projects, we're happy to have a discussion with you around that and try and point you in the right direction if it's not to be working with us. So we are on to the case studies at this point, which is, I suppose, some of the more interesting part of the day. So I'm going to stop sharing at this stage. And so the way that this part of the session will go is that we will have 3 different company case studies. We'll have both the company giving us a presentation about them and the problem and so on. And then the researcher from Shannon ABC, who is responsible for the work that was delivered, will also give a few slides on how the work was practically delivered within Shannon ABC. So the running order will be Deerland R&D will be first, Alahi Seafood will be second, and then SPV Healthcare will be third. So with that in mind, John, if you wouldn't mind sharing your screen with your presentation, and you can get started.
Thank you, Tim. I appreciate it. So my name is John Deaton, and I will start the presentation in presentation mode. There we go. So I'm Vice President of Science and Technology here at Deerland, Probiotics and Enzymes. Our main headquarters is in the United States. We're actually in Kennesaw, Georgia, just north of Atlanta. We actually have 2 facilities at that location, one an enzyme facility, and then the other one a probiotic facility that handles spores as well as non-spores. We are a supplement company. We formulate with many different probiotics and enzymes, and we develop our own innovative technologies as well. Where there's gaps in the market, we develop probiotic, prebiotic, enzyme-based formulations, And we have several different locations that we do research, as well as just facilities. We have several in the United States, but we also have others internationally, both in Denmark as well as Ireland. We do a lot of research. And
sorry, John, just to disturb you there for 1 second. I don't think your slides are moving on. They may not.
Sorry.
If you've got two screens, that might be the problem, or it may not be in presentation mode.
Let me try again.
You can scroll through them like that if you want to. There's no problem there.
Okay, just make it easier. Sorry about that.
No problem.
So we actually have several locations, again, several in the United States, others in Denmark and Ireland, where we do a lot of research. We do a lot of collaborations with universities and other institutions. We basically we research everything from in vitro to in vivo. Our purpose is actually to find solutions, supplemental solutions to help people in their daily lives, feel better, be able to do more, be able to be more active, etc. All of our technologies are around probiotics and enzymes, of course, and then we, of course, are responsible for the customization. We do the product technology as well as the education, both with our customers as well as the end consumers. We are a B2B company, but we do research that ends up educating the consumers in terms of how our products can help them. One of our products, one of the things that we've looked at for a long time is a enzyme-based antioxidant formulation. Free radicals are everywhere. They damage your tissues, skin, internal organs, etc. It's part of the aging process. It gives you wrinkles and basically can cause various issues as you get older. And so we wanted to have a very efficacious product. We wanted it to be enzyme driven so that you can have a small amount that gives you a strong activity. And so that's the reason that we actually pulled from chloroplasts. So we use spinach, we use an extract, we use enzymes to isolate that. We remove the cell wall because your body can't digest that. We also do certain techniques to actually stabilize as well as enhance the chloroplast contents so that we keep the glutathione pathway, which is what we're most interested in, intact. And so we can detect all these components in it. And then, of course, the glutathione pathway is Mother Nature's best and strongest antioxidant pathway. Every organism has it. And it helps eliminate free radicals in many different forms and areas, tissues, and liquids, as well as handling the different forms, hydroxyls, peroxyls, singlets, and superoxides. And then, but one of the things is, of course, we want to show the how it actually helps directly. We know that you get a lot of free radicals from many different areas, from stress, pollution, poor diet, etc. But we really wanted to narrow this down and actually show real life situations. And that's where we turned to Shannon ABC and collaborated with them on a project for this product. And I'll turn it over to Shannon to go through that.
That's great. Thanks very much, John. So if you can take your presentation off share, and then Neil, if you want to start away there. And thanks very much, John, for setting the scene for that.
So can you see my screen okay?
Yeah.
Okay, thanks very much. And thanks, John. So my name is Neil Burke, and I'm a business development scientist and project manager working for Shannon ABC, primarily at MTU Kerry. And I'm just going to give you a brief overview of some of the R&D work that we carried out in collaboration with Deerland. So let's see if my slide will move. Okay, so a lot of the work that we carry out at MTU Kerry involves cell culture. And for those of you who mightn't be as familiar with that, it involves the removal and isolation of specific cell types from either an animal or a plant. And essentially then it means growing them artificially in the lab. So just to mention some of the advantages of using cell culture for R&D work. Firstly, they're an ethical and economical alternative to testing on humans or animals. And they're also very time efficient. So the cells typically tend to grow reasonably fast, and you can use them at scale. So it can be reasonably high throughput. As well as that, the cell culture approach allows the testing of multiple parameters and endpoints. And so you're getting a lot of detailed information for one experiment. And then I guess the real selling point of it is if someone has a product or wants to test a compound and their end target is a specific tissue, then this approach allows you to test that. So for example, if you're interested in looking for efficacy in muscle tissue, then we can isolate and work with muscle cells, and so you're getting specificity there. And so overall, cell culture is a dynamic biological model, and we use it routinely, as I mentioned. So just to give an example of an application for cell culture work, a company or a research product will have a research project even, sorry, would have a natural extract or an active ingredient of interest that they'd like to test. And so what we'll do is we'll take that and we'll bring it to these cultured cells. So each of these wells that are highlighted here would represent a population of a certain cell type. So for argument's sake, a muscle cell. And then you treat each individual well with a different concentration of your sample, and you'll have your respective and relevant controls in there as well. And from that, it allows us to measure a range of different endpoints. And just to mention a few, we can see the effect of extracts or compounds on the cells in relation to the cell viability, the cell growth or proliferation, how much free radicals or ROS is produced within the cell in response to it, or if it can reduce those levels, and if it can modulate things like inflammation or DNA damage. So in relation to our work with Deerland, John has mentioned the product SolarPlast that they're looking at. And as you can probably remember from your school biology, a plant cell has a cell wall, and John mentioned earlier that it's difficult for that to be, the contents of the plant cell to be available when the cell wall is intact. And so we helped characterize the preparation of the SolarPlast in order to remove the plant cell wall and to free up the protoplast and chloroplast that are within that, and they contain the bioactives of interest. So John mentioned about antioxidants and free radicals, and essentially what they do in the cell is they stabilize unstable compounds that can cause all sorts of cellular and tissue damage. And one in particular, as John highlighted, was the glutathione and the glutathione pathway. And as this was of a key interest to SolarPlast product, we were able to use a commercially available glutathione assay kit and test the levels of glutathione activity after the preparation of the SolarPlast, excuse me. Okay, so from that point, then we were able to focus on some of the key target tissues. And one of the applications for SolarPlast is for skin health. And if it was potentially to be used as a topical product, you can see here in the diagram, we have three kind of broadly categorized layers, common layers of the skin. And the outermost layer is known as the epidermis. And the epidermis is primarily made up of cells called keratinocytes. And so we have keratinocytes, and we're able to grow those keratinocytes in the lab and add our SolarPlast to the keratinocytes with different insults. So essentially, we're stressing the cells and causing the cell to increase their amount of free radicals or ROS levels. And then using this dye, which you don't need to know the name of, things in science never tend to be tip off the tongue, but essentially, once we add the dye to the cells, the dye is taken up by the cell, and the more ROS or free radicals that are present within the cell, the higher reading we get. So on the graph on the right is a representation of a treatment where you will see an increase in ROS. And so we were able to treat these cells, these skin cells, with an insult, as well as the product SolarPlast, and we observed that SolarPlast reduced the damage caused in relation to ROS and the free radicals. Again, so because SolarPlast is, well, it has lots of beneficial attributes, as well as kind of improving skin health. It's also been implicated with gut health, as Deerland have done a lot of work around this already. And so what we were able to do was to take the solar plast and process it through an artificial digestion protocol. And so essentially, we're trying to mimic what would happen if a consumer or customer is ingesting the product. And so what we do here is we subject the solar plast to the pH and typical enzymes and salts that will be present at each stage of ingestion and digestion. So we go through from when it's in the mouth and how the saliva acts on it, to when it goes into the stomach and then on into the intestines. And at that point then, after we processed it, artificially digested the product, we were able to test to see if it had retained its antioxidant capabilities. And so we got a readout here, and there's an example on the right of the different levels of antioxidant capabilities that you can have from different samples and well-known antioxidant sources. And so we then confirmed that after the artificial digestion, solar plast retained its antioxidant capabilities. And then again, just to kind of add to that information, another beneficial attribute of solar plast is that it can help the gut barrier. So we use a CACO2 cell line, which is a commonly used intestinal epithelial cell line. And so that can mimic what's happening in the intestines. And what we do is we grow the cells, as you see from left to right here. We grow them individually, and we develop them or differentiate them into a more tightly formed tissue, for want of a better expression. So it forms an intact layer, and it mimics the barrier that's present in the gut. And then it's a similar approach to what we have done in other assays. We can treat these cells with an insult, and we can then try and prevent the damage caused by adding in solar plast. And we can do that using electrical resistance. So we have a specialized instrument that passes an electrical current through the layer of cells. And essentially, the more intact the cells are, the more their electrical resistance will be recorded or measured. And so if the cells are damaged or disrupted, you get a drop off in that reading. And so we're able to use that approach to show that solar plast can protect that barrier after an insult. So just to conclude, we've characterized the optimal solar plast preparation in relation to the chloroplast for efficacy testing. We validated the retention of solar plast bioactivity after the different preparation protocols and the artificial digestion protocol. And we confirmed its antioxidant activity in both the skin and gut setting, and demonstrated cell barrier protection in the gut as well. And so all of this information really strengthens the existing data that Deerland have for their solar plast product. So thanks very much for listening.
That's great, Neil. Thanks very much for that. So we're going to move on to our second case study now, which will be Michael O'Neill. So Michael, I don't know if you want to pop up your presentation there.
Okay. Are we up?
Not yet.
Okay. I can see it.
Yeah, it's starting to come up there now, Michael.
Is it up there?
Yeah, perfect.
Okay, just okay. We're Alahi Seafood, basically. We trade as pure ocean algae, and we're an aquaculture-based biotechnology company. We create vertically integrated value chains from microalgae to marine resources. So we're very strong green credentials, as we're basing our products on a raw material that's 100% environmentally sustainable and ultimately carbon negative. And for the last 5 years, we focused our attention on developing sustainable macroalgae-based infrastructure and aquaculture licensing, which hopefully will position us at the forefront of Ireland's marine bioeconomy. We have several capacities, including seaweed hatchery, seaweed farming, primary processing, and we're developing our secondary processing capabilities. So I suppose for us, with the voucher, it's all about the question. And if you ask a very wide question, you're going to get a very broad answer. So for us, it was about honing in on exactly what we were looking to do and what we were actually trying to achieve. And sometimes that can be problematic. In our case, probably we had to be led a little bit. I think that's where the team at Shannon ABC were able to fine tune. So this is an example of the question we were looking at. We were looking to review pre-treatment methods of seaweed and subsequent liquid-based seaweed fertilizers. So again, without going through the question, really, it was very straightforward. We developed a very tight description, and I suppose we hoped that by doing that, that we would have developed a very tight answer. So what we were looking for, we decided to go with our innovation voucher. And again, the desk-based report. What we were looking for was a very clear question that we wanted answered. I suppose we wanted good timelines for the delivery of that research, good interaction between the company and the researchers, so that it wasn't a case of a once-off, that it was more a process by which we got to get the information that we were seeking. And at the end, a very comprehensive report, fully answering all the questions that we were asking. Again, in our case, we can't speak highly enough of Shannon ABC. The team at Shannon ABC spent a considerable amount of time teasing out that question, and that was really, really important to us, because I think we went in with a very broad scope. And again, what do you really, really want to understand here? What was the purpose? And they spent a lot of time, maybe more time than they should have in our case, just getting down to the nitty-gritty of what we were actually asking. And again, because of that, we would hope that we would have had a really defined report coming forward, which we did. So again, we'd agreed the question to be asked. We agreed the timelines. The timelines didn't deviate. The report was finished as it was supposed to be on time. And again, that's critical because, like I say, for us, it was a piece of research that was preliminary to something else that we were working on, and we needed to get that piece of work done. And that was done very much as it should have been. I suppose we had the opportunity to discuss the interim research during the course of the voucher. And I suppose this allowed for some element of fine tuning and possibly refocusing, but there wasn't, we spent a lot of time on the question itself at the beginning, so it wasn't really so much a refocusing for us, but it definitely, I suppose, allowed us to build a relationship with the research team. And the final report, extremely comprehensive, it provided us with all the critical information that we required. The researchers were completely familiar with the information as well as the context, which we felt was very, very important. And they were able to give us key insights into the information, how best to interpret the results. Again, we would have been looking at how best to develop that information and bring it forward. And again, there was key insights that allowed us to maximize the results that we got from this. Again, we're very happy to recommend Shannon ABC, and we have. And we also see the opportunity to develop further research through Shannon ABC, through Tim and his team, and look forward to working with them in the future. Thank you.
That's great, Michael. Really appreciate that. Thanks very much for that. I'll just hand you over to Elva now.
Hi, Tim, can you see my screen?
I can see your screen. It's not on yet. Perfect. Yeah, that's working away now.
Is that working for you?
Yeah.
Great, great. So good afternoon, everyone. I'm Elva O'Connor, and I'm a Senior Business Development Scientist at Shannon ABC. And today I would like to speak to you about desk-based studies and what Shannon ABC can provide within an innovation voucher for a desk-based study. So desk-based research, I suppose as its name suggests very simply, is the type of research that can be performed over a desk. This is where a researcher finds, collects, analyzes, and reviews publicly available data about the research topic in question, and we can do this via the internet, peer-reviewed scientific journals, review articles, technical reports, academic search engines, and databases. So why are desk-based studies a good option for companies? So what do we need to know? So firstly, so the companies can see what has been done in relation to the product or service. This can really help to establish a broad understanding of the research field. And once a company has understood the research field, this can help with important decision-making and can also help to justify and plan for future work. For example, if there's a lot of data available about a specific product and the results are not promising, then it would be worthwhile to change something in, for example, the formulation, combine the product with different ingredients, or find an alternative use for that product or ingredients. Desk-based studies are also a good option for companies to create effective marketing plans for their products and services. So what information can come out of a desk-based study? So gathering information from scientific articles can tell us the different use, sorry, the wrong slide there, apologies. So what information can come out of these desk-based studies? So gathering information from scientific articles can tell us the different uses of a specific product or an ingredient and what methods are used to analyze the product. So marketing analysis can provide information on the products currently available on the market, the prices and the market size. Data on suppliers. So sometimes companies, for example, need a specific product to achieve their goal. An example would be if a company needed a source of spent grain. So a researcher involved in the desk-based studies would then contact the Irish breweries to directly speak to them about that. Patents, innovation disclosure information, innovation related statistics, or looking at the number of inventions in the field. Legislation review, so information on substances allowed in a product. So for example, in a food supplement and their concentration levels. We could also look at an example of permissible concentrations of heavy metals in animal food and in soil fertilizers in accordance with EU law. So what are the benefits of an innovation voucher? So as Tim mentioned earlier, innovation vouchers, they're funded by Enterprise Ireland to the value of €5,000, and they're there to support SMEs in new product process development, or to explore a new business opportunity, or to look at a problem that a company might face. So within this innovation voucher, we want to see what can Shannon ABC provide us. So we can provide sector-specific research expertise. So as Tim mentioned, we work with a wide range of sectors. So we work with the biotech, the cosmetic, the food and drink, pharmaceutical industries, and we have this research expertise across both our co-located facilities. So providing that sector-specific support, process troubleshooting, and a review of existing know-how and its adaptation to the client is a lot of what we can provide. And we'll look at the regulatory requirements also and provide a final report with the information collected and details of the outcomes achieved. So finally, we'll look at how we perform a desk-based study at Shannon ABC. So our researcher would write up a detailed report, and this would look at the aim of the study. So we will look at what was the question that we were asked. So what were the company's needs and what it was that we were actually hoping to achieve. So we worked with Michael O'Neill in the case of Alias Seafood. And this was to provide, as Michael had explained in his previous presentation, a consolidated report on extraction methods for liquid-based seaweed. And the report was derived by the researcher by reviewing the relevant literature. And the company had specifically asked for their interest in gaining information on various extraction methods available for wet seaweed. So in this report, we will review a research plan. So what the research methodologies, the tools might be to use to answer the questions and the needs for the company. And we will detail how we are actually conducting the research. So how we are collecting the information with and give relevant references and regulatory requirements. So with Ali's Seafood Limited, we conducted a scientific literature analysis looking at the various extractive methods, where we looked at detailed information about extraction protocols and the advantages and disadvantages of each technique, the information about, say, the mode of action of the different seaweed species, and also we looked at the marketing analysis. So looking at what seaweed products are available on the market, and looking at their brand names, their composition, and any invention-related database analysis. So looking at patented methods of fertilizers productions. So during this research analysis, we will make sure that we have covered enough data to answer to make sure we've answered the research questions, and that we've all the relevant information. So we will include graphs and tables into this report. And finally, the conclusion will detail the key points and the significant results. So in this case, we looked at the selection of the best methods of extraction that would help the company in achieving their goals. So I think as well for desk-based, just to point out, we will be showing now in our showcase video, we worked with Linus Life, and we started off with that client on a desk-based feasibility study. And we have gone on to work on a large innovation partnership, and that will be gone through in our showcase video. So thank you for your time, everyone. And I hope that's given you an overview of desk-based studies and what we can do and what we can provide you with Shannon ABC.
That's great. Thanks very much, Elva. So just as you're taking it off screen share, Stephen, if you can be getting ready. So final industry case study of the afternoon with SPV Healthcare first, Stephen, and then Elena, who'll talk more about the technical aspects in Shannon ABC. So Stephen, I'll hand over to you there.
Hello, everybody. My name is Stephen, and I work with a company called SPV Healthcare, and we have collaborated with Shannon ABC over a couple of years now, and with a number of different projects. So a little bit of background, I suppose, to SPV. We are a company based in Clonmel. We have a laboratory down there. We take healthcare products, whether it be cosmetic, medical devices, medicines, or food supplements, and we would take them from concept through to market launch, and maybe all of the different steps in between that. So there could be a concept, or we might need to look at what might be in the formulation, and what are the clinical outcomes, then look at what registration might be needed. If it's like it's a medicine or a medical device, does it need to be registered with the authorities? If it's a cosmetic, that's a different registration process. We would look at any intellectual property or so as well, and maybe like file some patents or do some prior art research, and then take it through to manufacture as well. And we do these with both products for ourselves, for SPV Healthcare, and also products then for other companies. So with Shannon ABC, what have we done? We have worked with them on a few different projects. Some of them are here. One of them was to grow a specialist microbial organism, a fungus, which was not available from any supplier or so, and it took us quite a bit of while to track that down. And so what we did was we needed to buy some freeze-dried culture, and then to look to incubate that and grow that and keep that then as a supply of that fungus. And that we were able to do with Shannon ABC. We have also done some cell line work and some immune profiling, so looking at different cell lines, looking at the immune responses coming from those. And we have also done some analytical testing and method development. So we have a product that we're looking to analyze some of the active in the product. And it is that that particular product that I'm going to talk about. But why did we choose Shannon ABC, and why work with them? There are a couple of reasons. They have quite a broad range of knowledge and specialist knowledge. They work in quite a range of areas, as Tim is saying, and that can be quite beneficial because it can be interdisciplinary work involved. And the range of equipment that's there and the type of equipment is really helpful and quite extensive. And then we can also negotiate and agree into intellectual property terms. And we have done our studies. Most of our studies have been done by the direct funding method, which we talk with the intellectual property as well. So this then the particular project that we talk about now, which is an analytical project problem that we have. We have a gel, which is an aqueous matrix in a polymer matrix, and there are two active ingredients in that matrix which we want to measure their dissolution, how they come out of the gel over a period of time, which was 3 days, and also over a number of pH values, so a number of chemical environments. Because especially in the early stages, the release profile is low, that the limit of detection was low, so that potentially should be aware of. And we had done some method development ourselves, but we hadn't been that successful with it, as we'd like to be. And we got at most maybe about 60% recovery. So of the active that were there, we were only maybe able to recover 60% of them. And so there was a problem with our method as well. So we went with that then to Shannon, and there with those points, and Shannon then was able to Shannon ABC was then able to develop a method, tweak the method, and develop it further, so that we actually got 100% recovery. So we knew then that all of the active ingredients that were there, that we were actually able to recover them. Were they coming out of the matrix, or are they not? And we were able to get full recovery. So we were able to consolidate all of the ingredient, the amount of actives that were there. Because the LOD limit of detection was an issue, we used mass spec, so HPLC mass spec, so that improved our detection, and we were comfortable able to then measure all of the actives coming out, even in the early stages when they were relatively dilute. And then, because this was not a trial, there were a few different ways parameters that had to be investigated, had to be looked at. That the study design that we put in place, and we did it over a few different studies, was flexible design. So there, as answers were got during it, we were able to adapt and cover those in, not necessarily almost in real time. And so that meant that the project was more adaptable, and it was also more efficient. So the results that we're getting are quite good, and we're really happy with them, and so we were quite happy to work with Shannon. So that's my discussion then, and I'll maybe stop sharing. And
that's great, Stephen. Thanks very much for that.
Okay.
And so just hand over to Lena now, who will talk to the kind of, so the practical expression of that in the lab.
Hi, everyone. My name is Lena Madden. I'm going to try to share the screen now. Tim, is it visible?
Yeah, that's perfect. And if you pop that on presentation mode, that'd be super.
Yeah, I'm going to try to do it now. So hi everyone, my name is Lena Madden. I'm a business development scientist at Shannon ABC in Limerick. And my main specialization is analytical chemistry and these few little babies. This is our new buy, 3-bed semi in Limerick, is a triple quadrupole MS detector. Which we use for very precise and very low level of quantitation, but not only that one, it has DAG detector on it that can work as a regular HPLC. This is our quadrupole time of flight, which we use for the untargeted analysis identification, what compounds are there. FTIR, which we use for fingerprint analysis, and that could be hooked up to the gas chromatograph and do really miracles, and that one will be the only in country. As Tim showed, HPTLC and GCMS chromatogram. And these instruments we use on routinely daily basis to conduct our studies. So we use the LCMS for analysis of the sample for the profile, for the solution profile of the gel that in question that we work with SPV Healthcare on. And when the guys came to us, they had the problem, as Stephen said, with the detection and sensitivity of the method. So the elution profile was quite problematic, and we believe that was because of the level of detection. So first of all, what we did, we have improved level of detection on instruments. So it went from way below level of limit of detection to very strong to the very, very strong signal that we could comprehensively quantify everything that is coming out of the solution vessel. So, and after that, we looked at the time of dissolution profile. So that's very important that when you, when the human being take this gel, that the compounds, the APIs are released gradually over time. We looked at the pH because each individual person have their own range of pHs, and dependent on how healthy they are or how the illness is progressing, the gel would have to work at different pH levels. So we looked at this efficiency of the release at different pHs. Then we look at the compound stability, because many compounds, well, could be very valuable when kept at temperature of human body or 37 degrees Celsius. They start slowly degrading or decomposing, and then do not produce the necessary effects. So we looked at the compound stability and have shown that it all works, the profile is working, and the compounds are stable. And also we looked into the solution medium improvement to bring it as closely as possible and simulate the bodily fluids of the human body for registration and for regulatory approval when Stephen needs to apply for that. So how different our approach to approaches maybe of normal regular research lab? We always ensure repeatability. We will not submit our report unless we know that our results are repeatable and they're the same at every time. We study statistics on the profiles, on the dissolution profiles for Stephen, and also for all our clients, for making sure that it works every time. We have inbuilt quality control. So we're working with the European Pharmacopoeia methods. We're working with the with all the FDA and European Medical Agency directives in order to improve quality of results coming out from our lab continuously. And the test results and methods that we produce are ready to transfer. So when I'm going to be giving Steven the results, it's not only going to be what we have achieved, it's also going to be how he can use the methods in his own laboratory. Okay, and how can he replicate? And the technology transfer is another big part of our day-to-day operation. So in every client, every company that comes to our lab, we take an individual approach. We are flexible in funding mechanisms, so we can advise, as team has explained, either to go to Enterprise Ireland for the sponsorship of the research funding, or we could advise if the company needs only very small, it has a very small question and it would be answered very quickly and easily. They could fund the projects themselves, or if they have enough capability. First of all, then we review their own processes. They will look into the process and the project and why the necessity of research have risen from the outset, okay, and maybe what approaches we can take to solve, or maybe the company might think that the solution would be, and that happens quite often, that solution could be in one way, but we could bring our innovative solution to the play and help the company to project develop and business develop or method develop for any application, for anyone and any application that comes to our door. And also we always ensure there is no client is too big or too small. We always pay the same attention and produce the same quality of the result, either from small and starting companies or for the big multinational clients that we have. And that is all from me. Thank you very much.
That's great, Lena. Thanks very much. So just as we're getting ready to start the Q&A shortly, before we do that, Lena, if you could take that off screen share, if that's okay. Thanks very much. So just before we do that, what we're going to do is show our promotional video. And just while I'm putting that up, just a big thanks to Elva, who's responsible for putting most of this together, and also the wider Shannon ABC team, who are very happy to kind of take part in the video as well. So just take a second for it to load up, and then I'll press play. So hopefully it'll load now. Here we go.
Tim, is the sound working?
Hold on a second, sorry. You're not hearing the sound.
I can't hear the sound.
Apologies, give me 1 second and I'll get that working.
No problem.
There's
Most
always
important
something. Yeah, the most important bit. Here we are, this should be okay now.
I'll let you know.
Yeah, do. Now, this should be okay.
We're good, Tim.
Shannon ABC is a collaboration between Munster Technological University and Technological University of Shannon, Midlands and Midwest. This co-location allows our industry and research partners to access the joint facilities and expertise across both sites through just one door. Shannon ABC is core funded as a technology gateway by Enterprise Ireland, and we work with companies all over Ireland to help them solve different scientific and technical challenges. We help companies get more value from bioresources. Whether this be as a raw material or a byproduct, we can help companies add value to their products and processes. We work with companies across a wide range of sectors, including food and drink, biotechnology, cosmetics, nutraceutical, pharmaceutical, bioeconomy and healthcare.
Shannon ABC has 25 staff across both sites in Kerry and Limerick. Our diverse team is made up of principal investigators, scientists, postgraduate students and business development staff that are here to help Irish industry. Companies can come to us to leverage our research expertise, our specialist equipment and facilities with the aim of optimizing processes, developing new products, or addressing scientific challenges. The key aim of the Shannon ABC Technology Gateway is to assist in the continued prosperity of industry. We offer access to wide-ranging expertise and know-how, state-of-the-art equipment and facilities, and access to a wide variety of national and international funding programs. We can help companies to identify the most commonly used funding options and provide support to them when completing applications and with their proposals. Shannon ABC laboratories are customised and future-proofed for wide-ranging biotechnological and analytical capabilities, including dedicated research bench spaces, analytical instrumentation, raw material processing, HEPA-filtered microbial suite, cell culture suite, and fermentation facilities to assist companies to address specific challenges.
Over the past 10 years, PBC Biomed, based in Shannon, County Clare, has worked with numerous companies, from startups to multinationals, in accelerating medical innovation. The facilities, knowledge and expertise made available to us from both universities through Shannon ABC has been invaluable in progressing these projects forward. I would highly recommend Shannon ABC to anyone who is looking for a professional and proficient research partner.
So here at Business Life, we like to create products that add value to how people look, feel and live. So initially with Shannon ABC, we worked on a feasibility desktop study on an active raw material that was very core to our business. Shannon ABC surpassed our expectations in relation to the work carried out, and we went straight into a full-blown innovation project. It has always been a collaborative working experience with the Shannon ABC folks, and we look forward to many more opportunities into the future with them.
Shannon ABC is here to help Irish companies. The ultimate utilisation of our knowledge, capabilities and equipment is when we deliver projects for Irish companies. With our wide-ranging expertise and our strong track record in delivering for Irish companies, I would encourage you, whatever your research question, to contact Shannon ABC to see how we can support you. So that's our video. So we're very happy with that, and thanks again to Elva for organising that and directing that. So as we're coming towards the end of the session, if anybody has any questions, they want to put them in the chat box, and we'll be certainly happy to address any questions you might have. And so I might actually start off with one just for John, if you don't mind, just on the solar plast product. Is that primarily intended as a kind of an ingredient, or are you looking at kind of developing your own kind of final product from that?
It is an ingredient. We do use it in many formulations. Customers have started asking for it. Again, the research that from the collaboration that we did with you has been great, and we've gotten a lot of interest. But it's an ingredient for formulations.
Okay. Thanks for that. Just wondering if anybody else has any questions. You can raise your hand or put it in the chat box, should be fine as well. So I have a question up here. So we have an enterprise R&D innovation voucher, and we have one product, food project already start. How soon could it start? Timelines, ingredient suppliers already confirmed. So it depends what the project is to a certain extent, and that would be one for a bit of a discussion, I suppose. How soon could it start? So I suppose I refer to it as our holding pattern of projects. So we've got a number of researchers employed in Shannon ABC whose job it is specifically to deliver kind of innovation vouchers and contract research projects that are generally anywhere from a few days to a couple of months of work. And so there will be a time, so if you were to, so you have the innovation voucher already, there will be a timeline there associated with setting up a budget code. So a certain period of time we have to allow for that. And it really depends on what the project exactly is, and who we have to assign to it. So how soon could it start is a little bit of an open question until we determine what the project is. The timelines to completion, again, it really depends what the project is. So if you're looking at a shelf life study, for argument's sake, we can do an accelerated shelf life study and you can get a 6-month shelf life or indicative 6-month shelf life within a month and a half or 2 months or something like that. But if you want to screen for, you know, what the antioxidant level of your food ingredients are, then that's a different thing entirely, I suppose. So it really depends on what the project is will dictate what the timeline to completion is. And just mentioned there, you have your ingredients and suppliers already confirmed, which is super because it saves a lot of time and hassle that way. But so for Nigel there, it really depends what the project is. And so certainly happy to take that up offline after the meeting if you like, or if you want to give a bit more context, I can give a bit more specificity to the answer, I suppose. So does anybody else have any questions? And again, as I say, feel free to kind of raise your hand. We have another message coming here. Hold on a second. Oh, thank you. Appreciate it. No problem, Nigel. So any other questions just around, vouchers or timelines for application and like that? As I say, certainly with the vouchers, it's a very easy application process. And I'm sure if Nigel has a voucher already, he can probably attest to that. It's a very straightforward process for companies to apply for an innovation voucher. And again, we're very happy to support companies in doing that. If they want to send on a PDF of the application form once they're finished, we can have a read through that and just make sure that everything is kind of okay in it, I suppose. So I might just pop this off. Actually, before I stop share, I'll mention something else just in case anybody else wants to put together a question. One of our partner groups, the Circular Bioeconomy Cluster Southwest, have an event on next week, Waste to Value. So basically, based on what kind of Shannon ABC do in terms of looking at bioresources and trying to extract value for that, it's very much aligned to what we do. And certainly for anybody that's here on the call who may have an interest on that, that's on next week. And so you can either contact the Circular Bioeconomy Southwest Cluster. If you Google it, you'll find it quick enough, or the email address is there as well. So info@cbcsw.ie. So I'm going to pop this off, stop share just to bring up the screen again. So I suppose, as I say, just as we're coming towards the end, just if there's any other questions.
Or if people aren't comfortable asking questions today, they can go to the Contact Us page of the Shannon ABC website, and or most people, attendees today, have my email address. If you would like to email me, I'd be happy to help.
That's great. Thanks for that, Elba. So look, we seem not to be getting any more questions in, which is fine. Obviously, all our presentations were very informative and answered all the questions. So I'd like to thank all the industry partners for giving their time today. It's much appreciated, and also all the Shannon ABC team as well for their input today. And as Elba said, if you've got any other questions after this, please do just check out the website and use the Contact Us link there. So thanks everyone. Everybody gets to leave class 15 minutes early and hopefully enjoy the nice afternoon that's out there. Thanks very much, everyone.
Thanks, Tim.
Thank you.
Great day.
Thanks, Tim.
Thanks, Tim.
Thank you.
Bye.
Bye-bye.
Thank you.